Hello students, today I will be discussing about DNA replication. So, DNA replication is a very vital topic. First of all, what is DNA replication? So, DNA replication is a process by which copies of DNA are being produced. Okay. So, write the heading. Let's. Okay. So, the definition is it is a process by which copies of DNA molecules are produced from a parental DNA. So, from parental DNA, when the copies of DNAs are being produced, exact copies, just like a Xerox copy, okay, that will be known as what? DNA replication. So, why is DNA replication necessary? Okay, for growth. Suppose few years back your height was a little less, okay, but now your height has increased, okay. Why? Because the cells grow. So when the cell grows, suppose this is a cell, from one cell two cells are produced. Not only cells but the DNA inside the cells are also produced, and DNA from the parental cell to the daughter cells, the DNA is copied. This process of copying the DNA is known as DNA replication. Okay, may it be growth, may it be heredity, may it be repair. In every process, replication is necessary. Clear? So, you will have to be clear that in DNA replication, from a parental DNA to daughter cells with the same exact DNA are being produced. Okay, means they are totally same. There is no difference. That's why. That's why if you take any of your body cell, if it have the same DNA, okay. Now, how this critical process occurs? Firstly, you will have to know where it occurs. Obviously, we will have to first discuss the basics of cell division. Okay. In cell division, we know from a, suppose this is a single cell, from that two cells are being produced, we know, and in this cell there is a DNA, and in the daughter cells also we will have the DNA. Okay. So, from a single cell to two cells, this process is known as cell division. And in cell division, there are few stages. That stages are G1, S, G2, and M phase. Okay, these are the four stages from which what the new cell is produced. So, we will not talk about the G1, G2 or M phase. We will be concerned about the S phase only. Why? Because in this S phase, only the DNA replication occurs. Okay. Now, how it occurs, how it takes place. That is very important. Suppose in case of prokaryotes, we know in case of prokaryotes there is no nucleus. So the DNA replication takes place where? In the cytoplasm. And in place of eukaryotes, the DNA replication takes place in the nucleus. Okay. In our syllabus or curriculum, we will be focusing mainly in the prokaryotes. Okay. So now let's see. Suppose this is a double-stranded DNA molecule. This is its 3' end, this is its 5' end. Okay. So DNA replication takes place from a specific region. Okay, means replication starts from a specific region. That region is known as origin of replication. Or ODC. Okay, origin of replication or ODC. Why origin of replication? What happens is, in DNA we see this is a coil structure. Okay, means one strand is coiled over the another. So in this coil structure, firstly we will have to open the coiling. Okay, so the opening of the coiling, sorry for the interruption. For the opening of the coiling, what we need is an enzyme known as DNA Helicase. Okay. What DNA Helicase does is it unwinds the DNA. Okay. The DNA helicase unwinds this DNA, and this DNA helicase, the DNA helicase knows few sequence of the origin of replication, means the ODC region. Okay, there are few sequences over here. Suppose A T G C C C A T A T A A T, like sequences like that are there, which is identified by which this enzyme DNA helicase. And DNA helicase, what it does, it unwinds the DNA. Okay, so this was the first thing, and this process is known as, we are talking about initiation of replication. Okay, this is initiation of replication. Clear? Now secondly, DNA replication cannot be started by its own enzyme. DNA polymerase cannot start DNA replication. So what will happen? Obviously, there may be some other enzyme. The other enzyme which starts DNA replication after this DNA helicase unwinds the DNA is known as primase. Okay. So just look. Suppose this is the unwinded DNA. Okay. this place has been unwinded by okay, this place has been unwinded by DNA helicase. Now this was the 3' end, this was the 5' end, and vice versa, this is 5' and this is 3'. Okay, now here will come RNA primer or primase. Okay, primase is nothing but RNA primer. Clear? This RNA primer adds few nucleotides of RNA. That is, RNA primer adds 5 to 10 nucleotides to start the DNA replication process. Okay, so the RNA primer adds 5 to 10 nucleotides of RNA primer. Okay, and this strand is known as the template strand. Clear? Now, after RNA primer has been added, then comes the enzyme named DNA Polymerase. Okay, this DNA polymerase, it gets attached here. This is the enzyme, and it adds the DNA, okay, or the nucleotides. Clear? Okay, so one thing we must know that DNA polymerase functions from 5' to 3'. You can see clearly, if this is the 5' end, if this is the 3' end, this will be the 5' end. So in this direction, 5' to 3' direction, what happens? The DNA replication or the DNA polymerase can add nucleotides. It adds DNA, not adds DNA. It adds DNA nucleotides. Clear? It adds DNA nucleotides. So this was the thing. So when the DNA polymerase comes into action, the elongation, this was the initiation stage till what? DNA helicase and RNA primer adding. But when the DNA polymerase comes in the picture, please wait. The process of initiation of replication comes into play. Okay, so DNA replication has started. So this strand is known as the leading strand. Okay, this strand, the red one. Okay, the leading strand, and the older yellow strand, which I have drawn in yellow, is known as the template strand, the 3' to 5' one. Okay, both are the parental strands, but the yellow 3' to 5' is the template strand. Okay, now Okay. Now what happens? This is the opposite or the lagging strand. Another with lagging strand I am drawing. Just watch. Okay. So in the opposite direction, synthesis will occur, but not in the same way, as because the this is the 3' end and this is the 5' end. Okay, so the RNA primer will come over here. RNA primer will come over here and add few nucleotides. Then the DNA polymerase will come and continue in this direction, but not the whole bubble. Okay, so in this place, what will happen? Few gaps will be formed. Okay, suppose this place it comes and adds like this. There will be some gaps. Okay, and this will be known as the lagging strand. Clear? This will be known as the lagging strand. Why? Because this is the opposite of 5' to 3' direction. Okay, and we already knew that DNA polymerase can work from 5' to 3' synthesis. It can do. Okay. Now, for the termination, the maximum of the DNA replication is complete till now. Only few things are left. What? There is some RNA primer over here. Suppose this is the DNA molecule. This is the DNA molecule. Okay. But in the pink area, it is still RNA. This RNA is then this gaps. See, there is some gap between both the DNA. Okay. Gap in the lagging strand is there. I was telling about. So these gaps are filled by DNA ligase. So what does DNA ligase do? Gap sealing. Clear? And this is the termination of replication. Clear? So this process is known as the termination of replication. So you can get a clear picture that from two strands, we were seeing two strands at first, but after that what happens, we will get 4 strands. Okay, when the replication is complete. Okay, there were 1 strand, 2 strands. In this case, 2, 3 and 4. Okay, from 2 strands we get 2 extra copies of DNA and DNA replication is complete. And this type of replication where 2 strands are old and 2 strands are newly synthesized, is known as semi-conservative method of replication. Okay. So this is the end of DNA replication. Thank you for watching.